much, Fatima, for your presentation. Thank you and Patricia and the student staff for inviting me to participate at this very nice meeting. I don't work in pest control, so I will talk about some basic feature of blood sucking feature. So how hard is feeding on blood, but at the end I will talk to you a bit about our work, which is related with some applied aspects. So I work at the Research Institute of Insect Biology. This is in an institute, a small institute located in a nice campus here in Tours. We are about 90 people working on all different aspects of insect biology, from ecology, genetics. And I'm interested since many years ago in understanding the adaptation of arthropods to the haematophagous way of life. So feeding on blood is not an easy task. Many species of arthropods use it as unique or main food, but because it is rich in nutrients and energy contents, it's mostly sterile, except for the parasites of snakes. This make them the vectors of many diseases to human and animals. But it is not freely available in nature. It circulates into vessels hidden under the skin of well-blooded animals, which are usually more active, bigger than the insect. So as you can imagine, if they are detected, they are rapidly killed. So the selective pressures acting on this kind of insects is extremely strong. So to live a haematophagous life, you need to cope with many challenges. So first, shedding the habitat with hosts which are at the same time predators. So many of the hosts on which they feed, they turn into predators when they are detected and just eat them. They must identify and locate this host, these potential food sources. They should be able to recognize the less defensive one in order to minimize the risk of approaching them and to feed on them. They must approach at the right time in order to avoid being detected. They have to localize blood vessels either under the skin. They need to gather blood without being noticed. They must spend a lot of energy. It's a very energetic process to suck blood from a small vessel. They must ingest as much as possible in the shortest time in order to minimize the risk that the contact time would pose. And they have to cope with different forms of stress, including thermal, toxic, and oxidative stress, in addition to parasites. I don't work on parasites, but I have investigated all these aspects at different moments, different models, ranging from kissing bugs, mosquitoes, ticks, and even lice from fossil elephants. So one of the most important aspects of the moment of the life of the haematophagous is seeking for a host and getting its blood. Many research has been done because it seems to be a critical moment, and we know now that different stimuli, odors, heat, humidity, visual cues are important. This information is integrated in different modalities in order to inform the animal that is the right animal to be eaten. That they must be done at the right moment of the day, so the condition of the life are very important. And also there are many endogenous factors that make insects avoid feeding when it is not necessary. or when other physiological processes are running, like development, nutrition, reproduction, circadian clocks which modulate this behavior, experience, and of course, infection. So it is not granted that if you release stimuli in the environment, the mosquitoes or bugs will come in response to this stimuli. So two of the most important cues that they use for locating hosts and others and heat, because the vertebrate animals, one-blooded animals are few sources of volatiles and also emits huge amounts of heat. So according to the degree of association with host, they need to invest much effort and sensory tools in order to get access to the food. So what you see here are different examples of blood-sucking insects, which establish different degrees of proximity with the vertebrate host. So lice live on the host, bedbugs with proximity, kissing bugs in the home, and finally mosquitoes with fly in the nature, and they can approach the host from a certain distance when they need it. So this degree of association is correlated with the sensory investment, with the sensory machinery that they need to devote to this activity. So there is an increased number of sensilla and sensory genes devoted to this tag when we see, we consider species that live less associated with the host. I say that odors are very important because we are huge source of odors. Our skin and the associated flora release several hundreds of volatiles, more than 350 have been identified a long time ago, and from them, each species will sense those that provide precise information about the location and the identity of the host, because the association between haematophagous and host can be very specific. So all this recognition is mostly associated to the other blend that is specific of each of the species. As Emmanuel said, these odors are sensed by specific sensilla, being molecular receptors specific for the recognition of these odors. And without surprise, so the same correlation that we had for other sensory features, the number and the type of sensory genes increases in species which show a further association with the host as mosquitoes. Many of these are other receptors that are involved in thermoreceptors, but this is mostly chemoreceptors that are expressed in these insects. Heat is very important cue, in particular for some species like kissing bugs or mosquitoes that are particularly sensitive to heat. In particular, kissing bugs are the animal with the highest thermal sensitivity. Now, at present, the ability to detect infrared radiation and to respond specifically to the temperature of the object. So in the recognition of the host are not only involved, but also temperature. So all these.
I'm sorry, Claudio, to interrupt. Sometimes we don't hear you very well. Maybe if you can speak a little bit closer to the microphone. I don't know if it could be better.
I'm sorry.
At least for me, that's how I hear you.
Now you hear. It's better now?
Yeah, maybe. Let's see.
Okay. So I say that heat is also a very relevant cue for the orientation from the recognition of the host, but also for locating blood vessels. As I said, blood circulates inside vessels hidden under the skin of the host, creating thermal heterogeneities that can be detected by haematophagous insects, and they can, this way, target the warmest areas when they bite. So heat is also important for locating blood vessels that they need to pierce. Here you see the ear of the mouse. This is the proboscis of the bag. This is the mouth parts that are inserted inside a small vessel. These are cells from the ear. And as you can see here, once the vessel is pierced, here you see a pumping activity. This movement of the vessel, okay, is not due to the heart, but to powerful pumps that are inside the head of the insect. So when they produce a wound, when they pierce the skin, our body and the body of animals, the skin reacts, triggering different processes aimed to avoid blood damage. The first damage, avoid bleeding. So they ingest the saliva, which composition is very complex, that is aimed to inhibit all these processes of vessel constriction, platelet aggregation, and coagulation that are triggered by the glue that the insect is producing on the skin. So once skin is pierced and they reach, sorry, this is in the head of the animals, the big part is reserved for powerful pumps which suck this blood. This is why they produce these pressure variations inside vessels, allowing the insect to inject this blood. This process that you see here, this is the wave of blood being ingested by the bag, is very costly. So the animal needs to invest a lot of energy to obtain this blood. As compared with the resting situation, the metabolism of the insect of the bag increased 17 times in order to be able to have a full meal. This energy, which is relatively big amount, is worth because the amount of energy they can recover from a single meal allow them living for a long time or even display an intense activity. If you see, this is the amount of time that an insect, female and male from a kissing bag can rest without food if it remains resting. This is the time for expending all the energy recovered by 150 milligrams of blood. If they move, when they disperse, this energy is enough for walking very long distances. So this is easy to understand why, despite all these constraints, hematophagy has appeared in many different species. So, as I said, the most advantageous is getting as much blood in the shortest possible time. This will space the contact with host in time. The events can be spaced for a long time, and also the time over the host reduces if the stamps are powerful enough to allow dodging in a few minutes. But these meals can be really good. Can reach many times the weight of the animal. For instance, in kissing bags, we can eat 10 or 12 times their own body weight in a matter of 10 or 15 minutes. Mosquitoes can be twice, twice, a bit more. But ingesting so much blood so quickly has several problems. One of the problems is that a lot of heat is entering in the insect body. You can imagine you drinking 100 liters of a tea at 50 degrees. 50 degrees is not a lethal temperature, but drinking so much tea makes a lot of heat entering quickly in your body. So the consequence of thermal stress will be dramatic. And haematophagous creatures are confronted to this problem at every feeding event. Here you see two Aedes aegypti, a female and a male. The female is feeding on my finger. And you can see in this thermographic image how she increases the body temperature. So they need to cope with thermal stress. To cope with thermal stress, hematophagous have developed different strategies. One is thermoregulation, as do malaria mosquitoes. They produce these small droplets during feeding, which contain urine and fresh blood. So they sacrifice a part of their food to produce this drop that allows them decreasing the temperature by evaporative cooling. So if you can see this video, once the female starts feeding, the temperature increases. But here, when the drop is released and kept attached to the abdomen, the temperature of the insect decreases. Soft ticks have similar strategy. I try to launch the video. Soft ticks produce a lot of urine that they release through coxal glands. This urine climbs onto the thick body by capillarity, because the vesicle is quite circulated, and by evaporative cooling, it succeeds in reducing the temperature. Video supposed to show this. I'm sorry, it doesn't work now. Other insects have found more sophisticated solution, like the kissing bugs, which is relatively bigger. Kissing bugs can ingest warm blood at near 40 degrees, but keep their abdomen close to the ambient temperature, thanks to a very efficient system of data current heat exchange in the posterior part of the head. So all the heat is retained in the head, is dissipated in the environment, but the blood arriving to the abdomen is at the same temperature as the head. So the bugs do not heat up when they feed on blood. This is good for avoiding thermal stress, but also for avoiding cannibalism, because, as I say, these bugs causes the highest thermal sensitivity known in animals. So heating up will result in cannibalism phenomenon, as you can see in these photos that have been provoked artificially. So, summarizing, haematophagous must cope with thermal stress, and they employ different strategies. Some of them use thermotolerance, so they support overheating and they repair the damage, and other thermoregulates. In the first case, the mechanism for tolerating this overheating is synthesizing quickly big amount of feature proteins. Thermoregulating haematophagous like Anopheles or kissing bags need just synthesize reduced amount of HSP, because most of the stress problem is solved through thermoregulation. This is not the only problem that they solve when they feed this big amount of blood. The other problem is the overload of toxic elements, because blood is composed mainly by proteins, and an excess of protein, in particular of certain amino acids, and the products of the ingestion of blood produce toxic substances in very high, when they are in very high quantity, and in particular the in group, which is a very reactive, oxygen reactive species that that results from the digestion of hemoglobin. So the specific degradation paths have been developed not only in haematophagous insect, but also in blood parasites, because both of them have the same problem. So finally, the other problem they are confronted to is, as I say, the accumulation of the M group, in particular when it is combined with light. So oxidative stress and photo damage by light have a synergic effect, increasing the oxidative stress in this insect. So they need to avoid light during digestion and of their food. So to finish, I say that I will present some applied aspect of the research we are conducting, and since a couple of years we are devoted to try to understand new repellents. So repellents are very useful for avoiding being eaten, and they are a great tool for personal protection to protect people from biting and from the transmission of pathogenic agents. But and they are known since many years ago. The first repellents really effective have been synthesized in the 1940s, but we still don't know how they work. At this moment, there is a problem. Synthetic repellents are very effective, but some of them start being targeted as producing toxic effect, as this, and natural repellents, which are supposed to be less toxic because they are natural. This is not completely true, but it's the general idea, are less effective. So for looking for new molecules is a priority in public health. But one major problem we have for guiding this research has been still do not understand why do repellents repel. Different hypotheses have been proposed, different molecular mechanisms have been proven, but there is not there is still not clear why, how they work, why they repel. So it's not rare to find this kind of title, a mysterious, enigmatic perception. So without knowing the action mechanism of repellents, in particular of DET, which is the gold standard of repellents, most of the search should be done by trial and error. This is the way many labs work. So in order to try to better understand the role, we asked the question, what does matter the most? The molecular action, which has been intensively studied since the last 20 years, or is the information repellent delivered to the insect? So in order to test this idea or try to solve this, we asked if it would be possible to make mosquitoes full of repellents, in particular, the gold standard of repellents. So for investigating this, we set up a classical Pavlovian conditioning experiment. Maybe you know the classic Pavlov experiments with dogs, when they made dogs associating the sound of a bell with food, and after some presentation of the association between food and the bell, the dog starts salivating in the presence of the bell. This is an usual method, and we tried to use the same principle for training mosquitoes. So we offer mosquitoes blood in an artificial feeder that you can see here, in which blood is the reward, heat is the unconditional stimulus, so the stimulus that will make mosquitoes die, and we associated food with the presence of a repellent. So if you do this several times, maybe that the animal associate both, and they did become into a predictor of a food. Then we tested the rest of mosquitoes to be only, it is supposed to repel naive insects. But we have seen is that actually mosquitoes did learn to like this. So this experiment requires a lot of controls. You need to present blood, be alone or associated that in different times, but not temporarily association, or pair at the same time. But we have seen that the association is possible, and these trained mosquitoes actually responded appetit-to this. So the balance of this can be switched from aversive to appetite. So in the next experiment, we do the same thing, but this time we presented mosquitoes with the hands of the person, one feet with it, and the other without it. And in this case, most of the mosquitoes choose the treated hand. So even in the presence of host stimuli, this can become unattractive for mosquitoes if the previous experience of the insects has been positive. The same for sugar. It's possible to train the mosquitoes to associate sugar plus this, and when you test mosquitoes to this, they become attractive. So, in conclusion of this part, This doesn't seem physiologically bad. It's not bad by itself. It's not the problem of chemical or molecular targets. That is the biological meaning of it that also matters. This makes them that they can learn to like it, to change this biological meaning, changing the value of repellents from appetite or aversive to appetite. So to finish, the other aspect that we are investigating concerning repellents in collaboration with Romina Barroso in Argentina is trying to, instead of trial error, trying to apply a rational way to found new repellents, and we focus in the gustatory system. So Emmanuel had already presented this, this particular system, and we are proving the concept using known substances that mosquitoes can taste, like caffeine or quinine. We characterize different reactions of mosquitoes to this compost, like contact repellency or feeding inhibition, and also the classical test for repellents. This is the in the cage where the repellent is associated, or the putative repellent is associated to the host. Caffeine doesn't seem to be very effective, like quinine showed a significant repellent effect on the IBCG. So summarizing, vertebrate blood is a unit or method for many arthropod species. Haematophagy is a risky and costly habit, yet has appeared many times. We don't know how many times, but at least 10 times in definite way in different evolutionary contexts along the evolutionary story of arthropods. And this has originated selective, strong selective pressure that make them to develop morphological, physiological, and behavioral adaptations in order to cope with all these challenges. And understanding this basic aspect of the biology of this particular way of life can help us to limit the transmission of pathogenic agents in the haematophagus. So I want to finish presenting just some of the people, the partners, and organisms that had supported this research in France, in Brazil, in Argentina, the United States, and also this would not have been possible without the support from the Studium, who hosted Marcos Pereira from the University of Belo Horizonte during a year in my lab, and also the program Sciences and Fronteras that allowed me to work with him and other colleagues in Brazil. So thank you very much for your attention. This is an old joke related with the song that was made all the time during my last visits to Brazil a long time ago. So thank you.